Hi everyone, we're back with another paper today. Today we're actually solving a biology paper after a very long time. We actually took a long hiatus from solving biology because it didn't get enough reach, but a lot of you have been requesting for bio papers. So this is actually 9700 paper 11 from the May June 2024 series, 1 hour 15 minutes. The time is 1 hour 15 minutes and it's paper 1 multiple choice biology, Cambridge AS level. So the ironic part is I am a doctor, but I don't solve much bio papers on the YouTube channel. But hopefully there will be a change from now on. Hopefully this gets as much reach as the physics or math videos. But before that, we have amazing news to share. The channel CIE Paper Solved is launching its first physical location starting from February 2025. The location is locked in. It's in Dhanmondi, Dhaka, Bangladesh. So if you're a student from Sunnydale, Green Hill Scholastic, or any other school, or even a private candidate from Bangladesh, please feel free to contact me. The WhatsApp number is given below. You can just call me directly as well. I'll reply, and you can sign up these forms. I'll be dropping a link for the forms in the description. We're launching two batches actually for the May June 2025 series. One will be a revision batch or a crash course covering all the content in a very compressed form and solving question papers. Batches of 4 friends joining together also get discounts. It's a monthly subscription for the crash course, and the mocks will be composed of 12 mocks, 10 to 12 mocks for each subject, like AS biology paper 1 and 2 combined, 12 mocks, A2 biology as well, which includes solution classes, and the crash courses and the solution classes, all of them will be recorded. So even if you can't attend, you will get the recordings later, so you can watch them if you've missed the class. Also, I'm also taking international students, so if you're from abroad, please feel free to join. You will have a separate batch. I'll have a separate batch for international students. So yeah, just fill up the form if you're interested and let me know. And I think we should begin. Let's not waste any more time and start the paper. So before that, let's take a look at the threshold, shall we? Where is it? It's over here. We are actually doing component 1-1. Okay, component 1-1. So it was at 33. It was at 33, all right. Okay, so let's start. Let's not waste any more time. A was at 33, B was at 30, C was at 26. Okay. So doing bio after a long time. Hopefully I'm not rusty. Let's see how it goes. So out of a total mark of 40, A was at 33, B was at 30, C was at 26, D was at 22, and E was at 19. I actually had the country highest mark for biology back in the day in Bangladesh in 2018. I got 95 out of 100. So what about A star? So as you can see, there's like a 3-4 mark gap, so it should be at 36 or 37. That is the mark for A star. So very hard to get, very little room for error, right? So let's not waste any more time and start. Okay. The diagram shows a stage micrometer scale viewed with an eyepiece graticule using a magnification of 200. Using the same magnification, a chloroplast is measured as 4 eyepiece graticule divisions long. So this is the stage micrometer and this is the eyepiece graticule. How long is the chloroplast? Okay, let's do this. So basically, as you can see, it's from 50 to 90. So 40 EPG units is basically 0.1 millimeter, right? 40 EPG units is 0.1 millimeters. So what about 4 EPG units? 0.1 by 40 times 4. 0.1 by 40 times 4, that gives you an answer of 0.01. Okay, so that's 0.01 millimeter. Now everything is given to micrometer. Let's convert this. So 0.01 millimeter is basically 0.01 into 10 to the power minus 3 meters. Okay, and that is basically 0.01 into 10 power minus 3 into 10 to the power plus 6 micrometer. Let's do this. 0.01 into 10 to the power minus 3 into 10 to the power plus 6, that gives us 10 micrometer, or 1 into 10 to the power 1 micrometer. Awesome. So the answer is A. Let's go to 2. Which range of cell diameters is typical for prokaryotic cells? We know we have eukaryotes and prokaryotes. So prokaryotes usually lie between this range, 1 into 10 to the power 3 nanometers to 5 micrometers. So the logic behind this is, look at how smart Cambridge is. Basically, a chloroplast was actually a prokaryote, but then it came to have a symbiotic relationship with eukaryotes. So as you can see that this size, like 10 micrometers, okay, the length of it, it's within this range. It's kind of within this range. But yeah, like mitochondria and then, you know, chloroplasts, all of them. So how much is this actually if you convert this to micrometers? Let's convert it. 1 into 10 to the power 3 into 10 to the power minus 9 times 10 to the power plus 6. That is actually a 1 micrometer to 5 microns. This is the usual length for prokaryotic cells. Although this is 10 micrometer, lies on the higher side, but that's okay. But this is what you need to have memorized. Okay, so this is the typical diameter of a prokaryotic cell. The diagram shows a typical animal cell. Where would nucleic acids be found? Interesting. Okay, so nucleic acids, right? So for 3, look, this is an animal cell. So 3 is a mitochondrion, so obviously it would have nucleic acid. We know that it has RNA. And on the other hand, 20, what is 20? It is the rough endoplasmic reticulum. It's the rough endoplasmic reticulum. So we know that nucleic acid is composed of either DNA or RNA. So yes, in the cytoplasm we can get a lot of RNA is found in the cytoplasm, nucleic acids, and in the rough endoplasmic reticulum you will also get you know, nucleic acids. Which structure is found in a typical bacterial cell? In a typical bacterial cell. Okay, so the answer here should actually be the template strand. Why not the other ones though? Introns and exons are not found in bacterial cells. We also do not have telomeres, because the, you know, telomeres are actually the you know, endpoints, right? They're the endpoints of the compressed chromosome. So we know that bacteria have you know, circular DNA. They don't have compressed chromosomes, so telomeres are not present. Introns and exons are not present. It's not that complicated for bacteria. And capsids are found in viruses. Okay, not. Okay, so what is the maximum number of hydrogen bonds that can form between a single water molecule and other water molecules? Okay, so it's H2O, right? H2O. So we have 2 lone pairs over here. So yes, this is del minus. This can form 2 hydrogen bonds because it has 2 lone pairs, and these hydrogens, okay, like with other water molecules. Check this. With other water molecules, these ones can form bonds. So in total we have 4. The answer for 5 is D. The diagram shows the structure of 4 amino acids in aqueous solution. Which 2 structures have an overall charge? Okay, so yeah, clearly this has a charge of +1 and this one has a charge of -1. Okay, but what is happening to the other ones? They are getting cancelled out. Plus minus is getting 0. So it's lysine and aspartic acid, aspartate basically. So 6 is going to be C, very easy. The table shows the number of carbon atoms and the number of carbon-carbon double bonds in the molecule of 3 triglycerides and the fatty acids they contain. The information is shown in the format number of carbon atoms to number of carbon-carbon double bonds. Which description matches fatty acid 2? Okay, of the 56:4 triglyceride identified in the table as X. Okay. So as you can see, it's the number of carbon double bonds in the molecule. And what is this ratio? It's the number of carbon atoms and number of carbon-carbon double bonds. So basically, first triglyceride has only one carbon-carbon double bond, only one of this, and in the one below there are 4. So clearly this has 2 carbon-carbon double bonds, because look, it has to add up to 3, it has to add up to 4, because here it's 1 plus 2, which is 3, so it must be 2 plus 2, which is 4. So since it has at least one carbon-carbon double bond, so obviously it is unsaturated. Okay, it is unsaturated because it has carbon-carbon double bonds. And what about the number of carbon atoms? Look at this. Like if you add this up, 18 plus 18 plus 15 is 51, right? But that's the fatty acid, because we know that a triglyceride is like this. It has C3 over here, right? And then it forms the ester bond, which is COO, because we have a fatty acid on this side, which is COOH. So basically we have the ester bond over here. So essentially the idea is, look at this, if we add this up, 18 plus 18 plus 18, that's 54. And here it was 51. So 51 plus 3 is 54, 54 plus 3 is 57. So look at this, same concept applies. So if you subtract 3 from here, what do you get? 56 minus 3 is 53. So the summation must be 53. So 17 plus x plus 18 is equal to 53. So 53 minus 17 minus 18, that gives you an answer of 18. So if you look at this for number 7, the answer must be 18 carbon atoms, and it must be unsaturated because it has 2 carbon-carbon double bonds. 8 collagen molecules are made up of 3 polypeptide chains interacting together. The individual polypeptide chains consist of a regular pattern of amino acids. Almost every third amino acid is glycine. Which protein structure of collagen are described? So we have a regular pattern of amino acids. That's the primary structure. So we have primary for sure. And if you have more than one chain, you are automatically talking about the quaternary structure, like collagen, and then, for hemoglobin, if you have 4 chains, if they talk about the 4 chains, so clearly the answer is B, because we have the regular pattern of amino acids, which is primary, and we have 3 polypeptide chains, which is quaternary. Okay. Next, the masses of the parts of hemoglobin are shown in the table. There are 1000 dalton in 1 kilodalton. Duh. What is the mass of the hemoglobin molecule in kilodaltons? Okay. So we know that a hemoglobin molecule has 2 alpha chains and 2 beta chains. Okay. So let's multiply that and add them up. So 15,126 into 2 plus 15,868 into 2. Now about the heme group. 1 molecule of hemoglobin has 4 heme groups for each chain. So we add 617 times 4 with that. So the addition comes to be 64456 daltons divided by 1K. That gives you 64.5 kilodaltons. Do we have that? Yeah. So 9 is D. Correct. What is the correct order of locations in the cell for the production of an extracellular enzyme? Okay. So the general idea is the Golgi body, the Golgi apparatus. Okay, the Golgi apparatus is mainly used for packing. Okay, so it should be at the end. The Golgi apparatus or Golgi body should be at the end Okay. Next, the idea is the production actually, transcription, then translation, it actually starts from the nucleus. So the start should be nucleus. So 10 is A. So it goes from the nucleus to the ribosome, rough endoplasmic reticulum for post-transcriptional modifications, right? Post-translational modifications, like, they add, there's glycosylation, there are others, and then we get Golgi body for packing. The graph shows the trend for an enzyme catalyzed reaction. Which labels are correct? Okay. So, yeah, they might be related to rate. So, why not A? A is wrong. They're saying that X is the rate and this is the substrate concentration. We know that it would plateau. Right? So yeah, that's why it's not the correct one. Enzyme concentration and temperature. Enzyme concentration and temp. No. Temperature is always, remember, it's the independent variable, so it can't be B. It can't be on the y-axis. So yeah, it's C definitely. C is the correct answer. Let's take a look why. Because, like, pH is independent and this is rate. This is the optimum pH, but after optimum pH, we know that it will denature, the enzyme will denature, and the rate of reaction will decrease. Yeah. Number 12. Which statements about the Michaelis-Menten constant are correct? So Michaelis-Menten constant actually determines the enzyme affinity. So the higher the Michaelis-Menten constant, the lower the affinity. That's the general idea. The higher the Km, the lower the affinity, or the lower the Km, the higher the affinity. Okay. So let's look at the statements. They're saying that the higher the Km, the higher the affinity. This is totally wrong. So 1 is wrong. Okay, so the answer has to be D. Km is a measure of the degree of affinity, right? And Km is defined as the substrate concentration at which the enzyme functions at half its maximum rate. This is also true. So the lower the Km, the higher the affinity. Number 13, the diagram shows how nicotine is transported from the blood plasma into cell using a type of co-transporter mechanism. In the phloem tissue, there is a co-transporter mechanism that moves sucrose into the cytoplasm of a companion cell. Which statement correctly describes a similarity between the co-transport of nicotine and the co-transport of sucrose? Okay. So to do this, we need to revise the co-transport of sucrose actually. Okay, let's talk about it. So what is the general idea? Okay, we know that, suppose this is a companion cell. So what we do is we load sucrose actively into the companion cell, and at the same time hydrogen is pumped out actively using ATP. And then a lot of hydrogen builds up actually, a lot of H plus builds up over here. So there's a co-transporter over here. So now the hydrogen will move back down the concentration gradient of hydrogen, since a lot of it is built up over here, and at the same time it moves through a co-transporter, so it will actually carry sucrose along with it. Okay, along with it, because it's a co-transporter against the concentration gradient of sucrose. So this is the same logic. Look, protons move down their concentration gradient. There will be a lot of protons over here, and at the same time nicotine is transported against its concentration gradient into the cell. So it's just that sucrose moves in the same direction for phloem, but here basically it's not the same direction, but we use the co-transporter for facilitated diffusion. This is why B is correct. Why are the others wrong though? The co-transport proteins generate a proton gradient by moving protons out of the cell by active transfer. No, this is done by another proton pump. That is completely separate. See, the protons move through the co-transport protein in the opposite direction to the movement of nicotine and sucrose. No. In sucrose actually moves in along with protons, but here nicotine moves out. Nicotine moves out and protons go in. So it's not the same. Here this is wrong because actually sucrose will move in the same direction. So C is wrong. The co-transfer proteins use energy. No, this does not use ATP. ATP is used initially to pump out protons, but not in the second stage. Question 14. The diagram shows a simple metabolic pathway. The letters W, X, Y, and Z represent 4 different substances. At each step in the diagram, the substrate undergoes a chemical reaction catalyzed by an enzyme. The reaction produces the next substance in the pathway. Which statements correctly describe the enzymes taking part in this metabolic pathway? Enzymes are globular proteins. We do know that. Okay, enzymes must be globular proteins. So 1 must be correct. We need 1, which is why D is wrong. An enzyme has to be a globular protein. And to be globular, you need to produce, hydrogen bonds. And also you need hydrogen atoms in your structure. You need to be hydrophilic to be a globular structure. So obviously you need to be attracted to water. So you need hydrogen atoms. So yeah, this must be correct. 3 must be correct. Now what about 2? They all have the same tertiary structure. We know that they are each catalyzed by an enzyme, a different enzyme. So having the same tertiary structure, it's actually the folding up of the secondary structure that produces the tertiary structure. The enzymes might have different tertiary structure. It's not a fixed thing. Okay, so 1 and 3 are correct, but 2 is wrong. Okay, because that's not possible having the same tertiary structure. Next. A student observed the effect of 2 different concentrations of salt solution on blood cells. The student added each concentration of salt solution to one of 2 microscope slides, and then a small drop of fresh blood was added. Each slide was viewed using the high power lens of a microscope, and the student's observations were recorded. In slide 1, no red blood cells were visible, and in slide 2, the red blood cells were visible but looked slightly crinkled. So what does crinkled mean? Like this. So water left the cells. So in slide 2, since they're crinkled, water left the cells. What does that mean? Initially, the water potential, psi, this is psi, the water potential was higher. It was more positive inside the cell and it was negative here. As a result, water left. So the psi of the cell was more negative. Wrong. The psi of the cell was more positive. The water potential of the external solution was more negative. Correct. Then the water potential inside the cell. As a result, water left. That's why it became crinkled. Okay, this looks good. There is a net movement of water out of the cell by osmosis. This also looks good. The water potential was similar but slightly more negative than that. No, the water potential of the cell was more positive than outside. Okay, let's look at these now. I'm going to ignore this because those are wrong. The water potential of the cell was more negative than the water potential of the external solution. Absolutely correct. Why were no red blood cells visible? Because look, if it's more negative than the one outside, then water will enter the cell and it will burst. So we will not see any red blood cells anymore. That's why 15B is correct. Okay. 16. Which statement correctly describes facilitated diffusion? Okay. So facilitated diffusion. What do we need for this? We need channel proteins or carrier proteins. Either works. It's not fixed. So only channel proteins is wrong. So it's channel proteins or carrier proteins, not only carrier. Both. It can work with both. Okay. So in a process that may or not may change shape. No. Energy is not required. It's actually a passive process. So B is correct, right? So learn this for paper 2. It's a process that occurs with channel proteins or carrier proteins in a process that may or not may change the shape of the protein. 17. Which row is correct for stem cells? Okay. So what is a stem cell? It can actually, differentiate to form another sort of cell. Okay. So this is very important. So remember, a stem cell cannot really repair cells. Okay, the stem cell cannot really repair cells. This is very important. It can differentiate to form new cells, but it cannot repair old cells. Can it be involved in the formation of phagocytes? Yes, because it can differentiate to form a WBC. Like we know that in bone marrow cancer, leukemia, in blood cancer, basically, WBC. We need bone marrow transplants to produce new stem cell therapy essentially. So yes, 17 is D. 18. Eukaryotic organisms grow and increase in size as a result of cell division. The diagram shows some stages of mitosis with one stage missing labeled X. What happens in stage X? So we know we have prophase, which is divided into early and late. We have metaphase, right? Then we have anaphase followed by telophase. So that is the general idea. So clearly this is in the equator, so it's metaphase, and there's one stage missing in the middle, and this is telophase clearly. So stage X is actually anaphase. What happens in stage X? So in metaphase they line up across the equator, and in stage X anaphase the contraction starts. The spindle fibers actually contract because the microtubules shorten and the centromeres divide. So yeah, this is for metaphase. Okay, so this is wrong. This is in prophase. Okay, and this is not even for here. This is not for here. This is for meiosis, which you'll learn in A2, where we actually get, homologous chromosomes actually line up. So the answer must be D. This is for the anaphase. So 18 is D. 19. The graph shows various distances measured taken from the start of metaphase of meiosis. The graphs are to scale when compared to one another, which correctly identifies the distance. So this is metaphase, right? They are lined up across the equator, and then they will start going to the opposite poles. So the spindle fibers contract, the microtubules shorten, and they will go towards the opposite side basically. And then in telophase, we'll start getting two different cells. So which row currently identifies the distance measurement for each graph? So X, for the first one, is distance between poles of spindle. Distance between poles of spindle. So if you think about this, is it actually correct? It stays constant and then decreases. The poles of the spindle, the poles are here. The poles are here. It doesn't really decrease. Okay, it doesn't really decrease. That's wrong. The distance between the poles does not decrease. So these two are wrong. There's no point C and B. We should focus on C and D actually. Okay, so if we check 19, C, distance of centromeres from poles to spindle. Okay, so as you can see, initially at the start of metaphase, the centromeres are located here, but afterwards, look, the centromeres move towards the poles. So yeah, this is correct because it actually decreases. So it's between C and D. And what about the other ones? Look, the distance between the poles of the spindle, there was initially a distance and then it increases, okay, because they are apart at metaphase. So that's why this is wrong. If there was a distance, it might have been correct. Distance between the sister chromatids. Initially, look, there was no gap between the sister chromatids, but then they split and moved towards opposite poles. So initially there is no distance because they are at the equator, they are close to each other, and then they split and the distance increases, which is why the answer is C. 20. Which way is correct for the start of anaphase of mitosis? So there will be 3 questions on chapter 5, mitosis basically. Start of anaphase. So when anaphase starts, what happens? They were at the equator, but now they split. The sister chromatids, they move towards the poles. Okay. So what is the form of DNA at the start of anaphase? They are no longer chromosomes. What has happened? They have turned into separated sister chromatids. Okay. DNA is associated with histone protein. Yes, they are still associated with histone proteins because they are still compressed. So it should be always, not sometimes. State of the cell surface membrane. It is intact. Okay, at the start of the cell membrane will always be intact. Come on. It's not the membrane of the nucleus. So it will always remain intact. If you want to see a diagram, I can show you guys. Look. But where's the course book here? Look, the cell membrane always remains intact. Okay, it's not broken apart. Cell membrane remains intact. You can take a look at this photo. It's from the course book. Moving on to 21. 4 bases. Which diagrams show thymine and cytosine? So we have A, T, C, G. Okay. So these are 4 bases. We have purines and pyrimidids. Purines and pyrimidids. So what do we know? The purines include adenine and guanine. Okay, the purines include adenine and guanine. They're double ring structures. Okay. So this is adenine. This is adenine. And this is guanine. Double ring structures. And there's also uracil for RNA. Thymine, cytosine, thymine, cytosine, and uracil. These are actually pyrimidines. Bigger name, but smaller ring. So let's identify those as well. You need to remember the structures actually, okay? So uracil isn't given here. But we'll look into cytosine and thymine. The trick about thymine is it actually has a CH3 bond outside. Look, it has a CH3 bond that's outside. So that's thymine. And for cytosine, there's just the double bond and an NH2 outside. That's it. So thymine essentially has 2 CO groups and a CH3. That's the striking characteristic. Uracil only has 2 CO groups and cytosine has 1 CO double bond, okay, and 1 NH2 outside, amine group outside. This is cytosine. So we are looking for cytosine and thymine. So we don't need that much details. We just need to locate the pyrimidines here, which are single ring structures. So that's clearly 2 and 3. 21 should be C, okay? The diagram shows the nucleotide sequence of a small section of the transcribed strand of a gene. The table shows the amino acids coded for by 10 mRNA codons. What is the sequence of the 4 amino acids in the polypeptide translated from this small section of a gene? Okay. This is the gene. Fine. So let's do this. So CGG. CGG. So what you need to understand is this has been transcribed. So when we do translation, we will actually get the opposite one for the mRNA codon. So we know that it's CG and AT. So we'll get GCC, CGG, GGC, GCC. So I'm looking for GCC. So the mRNA codon for that, because this is the gene, right? So the mRNA codon for that, the complementary one, I'm looking for GCC, ELA. It will start with ELA, alanine, and then CGG, arginine, ELA, then GGC. Let's look for GGC. That's gly. Okay, then GCC again. So we have ELA again. So I'm looking for ELA, gly, and then Li again. So yeah, I'm going for 22b. What does the process of translation require? So translation is basically the formation of a protein from the messenger RNA. So of course we need the mRNA. mRNA is a must. It's always there. And for translation we need ribosomes. That's a must. We need ribosomes. That is why these are gone. We need ribosomes. Okay. And of course, since we're making a protein, we need amino acids and tRNA, of course, okay, to bring the amino acids. And DNA polymerase is required for replication, and RNA polymerase is required for mRNA synthesis. 24. The electron micrograph shows a longitudinal section of a part of the stem of a plant. What is the name of the structure labeled X? So clearly I was talking about this, the companion cell and the phloem sieve tube element. So what is this essentially? If you take a look at it, I will show you. This is actually a phloem sieve tube element. Let's show you why. Let's look at the course book. Let's look at a few pictures from chapter 7. So this is what it looks like. We have the phloem sieve tube element. Do we have any other pictures? And the companion cell will be just beside it. The sieve tube element will have sieve plate, sieve pores, mitochondria, cellulose cell wall, cell membrane, cytoplasm. So sieve tube elements are elongated in shape. They are joined end to end vertically to form a continuous tube. It has a cell wall containing cellulose. However, the amount of cytoplasm is reduced and only forms a thin layer, and there is no nucleus, no ribosome. That's the characteristic feature. No nucleus and no ribosome. As you can see, there is no nucleus and no ribosome here, and yeah, you can see the sieve pores. So this is the phloem sieve tube element. Which substance makes xylem vessel walls impermeable to water? This is just memorization. Lignin is impermeable to water. The following tissues carry an electrical impulse during the cardiac cycle. In which order does electrical impulse start? So the sinoatrial node is known as the pacemaker of the heart. So it has to start from the sinoatrial node, okay? So it starts from the sinoatrial node in the right atrium, and then where will it go? It will go to the muscular wall of the atria at first, okay? Then what do we have? Then we have the AV node, the atrioventricular node, and then it will go to the bundle of His and the Purkinje fiber and spread throughout the heart, okay? So we are looking for sinoatrial node, muscle wall of atria, followed by the AV node, AVN. For 27, the graph shows the changes in the volumes of the ventricle during a single cardiac cycle, which is correct for the atrioventricular valve at P and for the semilunar valve at R. Okay, so you guys need to understand something for the cardiac cycle. This is very important. What does the systole correspond to? Ventricular contraction. So when the ventricle contracts, what essentially happens is the bicuspid and tricuspid, or the atrioventricular valves, will close and the semilunar valve will actually open. Okay? And what happens at the beginning of the diastole? If you think about it carefully, what does this mean? So at first, let's talk about the atrial systole. When the atrial systole occurs, what happens is that basically the atrioventricular valves will open. The atrioventricular valves open. Okay? And when the ventricular systole occurs, the atrioventricular valves will close and the semilunar valve opens. Okay, so that is exactly what will happen here. 27 is A. Why? Because at this point, the AV valve opens. And at this point, when the ventricles contract, what will happen? Suppose this is the heart, okay? So when the ventricle contracts, what will happen? This was open, right? Now this will close. At this point, the AV valve closes. And you know that we have the pulmonary trunk and the aorta over here, and the blood will rush through here and the semilunar valves will open. Okay, and the semilunar valve opens due to the contraction. That actually occurs here as well. These occur simultaneously. Okay, so like the closing of the atrioventricular valve and the opening of the semilunar valves happen at the same time. But when the diastole occurs, this part will relax now, right? This will relax, and at the same time, at this point again, the semilunar valve closes. Okay, so this is so important. Like at the beginning of the ventricular systole, the atrioventricular valves will close, and the semilunar valves will open. But at the end of the ventricular systole, the semilunar valve will close again. Okay, because the ventricles are relaxing now. That's the general idea. So 27 is A. 28, the graph shows how changes in oxygen concentration affect the percentage oxygen saturation of human hemoglobin and cat hemoglobin under normal physiological conditions. The partial pressure of carbon dioxide was kept constant at 5 kilopascal, and the temperature was kept constant at 37 degrees Celsius. Which conclusion is supported by the graph? So we know that the Bohr effect occurs, like whenever one oxygen binds with hemoglobin, the affinity for hemoglobin increases more and the graph shifts to the right. That's the Bohr effect. So a movement to the right means that you have a greater affinity. That's the general idea. Movement to the right means greater affinity for oxygen. Okay, that's the general idea. So let's take a look at this. Then we'll understand. So let's revise. The S-shaped curve is mainly due to the fact that when one oxygen binds with hemoglobin, it can take 4 molecules of O2, so for 8 atoms. So as more oxygen binds with hemoglobin, the affinity keeps on increasing progressively. And for, what is the Bohr shift? It is actually in the presence of CO2, in high CO2 partial pressure of CO2, in high CO2 concentration, hemoglobin is more likely to release the oxygen. So, more likely to release oxygen. So, it has less affinity for oxygen at high partial pressure of CO2. That's the Bohr effect. So in respiring muscles where there's a lot of CO2, we know that there will be a reaction catalyzed by carbonic anhydrase, so it will become acidic. So the hemoglobin actually has more affinity for acid H+, so forms hemoglobinic acid, and in the process of doing so, it releases oxygen. So if a graph lies more right to the right, what does that mean? That it has less affinity for oxygen because it's more likely to release it. So the affinity of cat hemoglobin for oxygen is greater than the affinity of human hemoglobin for oxygen. No, the more you are to the left, the more the affinity for oxygen. At high partial pressure of oxygen, cat hemoglobin picks up oxygen more easily than human hemoglobin. No. Here, look, if you lie to the left, okay, if you lie to the left, that means you pick it up more easily. But it is true that at low partial pressure of oxygen, the cat will release hemoglobin more faster because it has less affinity for hemoglobin. At low partial pressure of oxygen, oxygen is released more easily. This is correct. Okay? So we can say it like this, that at low partial pressure of oxygen, the cat is more likely to release the oxygen, and at high partial pressure of oxygen, the human hemoglobin is more likely to pick up oxygen. So that's the general idea. You can learn more from the course book from this page. How much is this? What's the page? I don't know, but chapter 8 basically. 208, page 207. The same logic is given here. It's very easy to understand. But basically on the right hand side, like if the graph is on the right, there is high carbon dioxide concentration or less affinity for oxygen. Okay. Moving on to 29. Which statement is correct about how oxygen combines with hemoglobin? Combining all 4 oxygen molecules with hemoglobin does not affect shape. Wrong. I just told you it affects shape, right? Okay. Next. One oxygen molecule can combine with each heme group, right? So there are 4 heme groups, so 4 oxygen molecules. How are alveoli adapted to their function? So a lot of things. High surface area, squamous epithelium to increase surface area, okay? So yeah, we have that. Contains squamous epithelium, short diffusion distance, and it also increases the surface area. And these are part of the bronchus, okay? 31. Diagram shows oxygen diffusing from the space inside an alveolus into the blood through the gaseous exchange. What would increase the rate of diffusion of oxygen from alveolus to the blood? Okay, so this should be high, this should be low, and the thickness should be low. Okay, so let's see which one matches. We need to increase x, basically increase x. So yeah, we have our answer. This is the only thing that can be increased, so 31 has to be D. Okay, more oxygen inside the alveolus means faster diffusion. Which tissues are present in bronchus? Very, very important. Okay, this is so important. So it contains all three. It contains ciliated epithelium along with smooth muscle, and also contains cartilage. But bronchioles do not contain cartilage. Okay, this is something you need to memorize, and there's, I think, a chapter in the book where you can memorize everything. Wait, let me show you. Yeah, this chapter here, you will have all the details for the trachea, bronchus, bronchioles here. So you only have trachea in the cartilage in the trachea and bronchus, and as well as goblet cells, but later on you do not have these. And smooth cells till the terminal bronchiole cilia as well. Okay, and gas exchange in the alveoli and alveolar duct. This table you need to memorize. All right. 33. Steep concentration gradients must be maintained for efficient gaseous exchange to occur in the human lungs. Which row correctly describes how steep concentration gradients can be maintained? So the elastic fibers in the walls of the alveoli recoil when breathing out. That's true. Continuous supply. This is very important. It should be A, both. Because look at this. If you get deoxygenated blood continuously in the pulmonary artery, what does that mean? The concentration of O2 will be low in the blood, so that creates a steep concentration gradient. And elastic fibers in the wall of the alveoli recoil. When breathing out, that means you quickly get rid of the deoxygenated blood. Sorry, you quickly get rid of the carbon dioxide. Okay, and get more oxygen back in. Because of the recoil, you get the oxygen out, you get the gas that you get from the blood, the one that contains carbon dioxide and less oxygen, it gets out faster, and then you can get oxygen in faster from outside, inspire more air. Okay. 34. A scientist investigated the effect of an antibiotic on the treatment of cholera. 320 people with cholera were divided into two groups. One group was treated with the antibiotic, while the other group was not given the antibiotic. Both groups were given fluids containing sugar and mineral salts, ORS. The scientists recorded the number of days that each person had diarrhea. The table shows the results. What is the percentage of decrease in the mean time that a person had diarrhea when they were treated with antibiotic? Okay, so 5.3 minus 3.2 divided by 5.3 into 100%. This is what it should be. 39.6. This is the percentage decrease. Okay, decrease divided by original, the one you're comparing with, then times 100%. How does the antibiotic penicillin affect the metabolism of a bacterial cell? So what does penicillin mainly do? It prevents the formation of the cross links. So holes are formed and it bursts. This is the only answer. There's no, don't bother wasting time with the other ones. It just prevents formation of cross links in the cell wall of the peptidoglycan molecule. Scientists studied the multi-drug resistant bacterial infections in children caused by one of the bacteria between 2007 and 2015. The percentage of multi-drug resistant infections rose from 0.2 to 1.5%. What was the percentage increase? Okay, so basically let's try doing this. 1.5 minus 0.2 divide by original into 100%. Let's see. 1.5 minus 0.2 divided by original into 100%. That's 650. Always divided by the original one. Okay, original 2007. Whooping cough is a highly infectious disease of the gas exchange system caused by the bacterium Bordetella pertussis. Which method provides protection to infants against whooping cough and reduces the chance of developing the disease later? Okay, so they need to be vaccinated basically. Okay, so because they said reduces the chances of developing this disease later, so we're talking about a vaccine. So what is a vaccine? It's basically injections of the antigen of the bacteria itself so that the infant gets exposure to that. And you know that in the EPI schedule, the expanded program of immunization, we have the penta vaccine, which consists of DPT, okay, then Haemophilus influenza B, so like 5 diseases there. So in deep, we also cover pertussis there. It's a vaccine where we use injections of antigens from the bacteria itself. Okay. The electron micrograph shows a type of blood cell. What can be concluded from this electron micrograph? Okay, so as you can see, there's a lot of rough endoplasmic reticulum. So it makes a lot of proteins. That's the general idea. Okay, because rough endoplasmic reticulum is required for protein synthesis translation. Why is mitosis important to the immune response? Okay, so mainly we need to produce more cells, more lymphocytes, more plasma cells, and so that they can produce antibodies. So the answer should logically be A, because B lymphocytes can use mitosis to divide to form more plasma cells. Recognition is not by mitosis, okay, and phagocytosis is not by mitosis either. And antibodies are proteins, so that's the work for rough endoplasmic reticulum. What is fused with B lymphocyte to form a hybridoma cell in monoclonal antibody production? Very important. It's the myeloma cell. The myeloma cell is fused with B lymphocyte to form the hybridoma cell. This is very important. This is also available in chapter 11 of the course book. So if you haven't read that already, please do, because this monoclonal antibody, it's a very hot topic right now. And yeah, it is actually very common right now in both, paper 2 as well as paper 1. At the whole part, it's in page 281 and 282. Here the whole process is mentioned. Sorry, it's loading. So how do we do it? Antigen is injected into the mouse, and then we get plasma cells, and then we combine it with cancer cells. Okay. We combine with cancer cells. These cancer cells are basically the myeloma cells. Okay, so that's the general idea. So yeah, it was overall an okayish paper, not much hard questions. Like maybe here and there were a few hard questions. But yeah, we'll be launching more bio papers soon, more paper 1, more paper 2, more paper 5 and 4, so don't worry. So if you want to see that content, please do remember to subscribe to the channel, like the video if you liked this content. And yeah, you can fill up the form if you're interested in joining classes if you're from Bangladesh or abroad. I'll be taking in more students. Quick shout out to my top tier patron for the month, Nigel. You can support me on Patreon if you want one-on-one video meetings with me over Google Meet, where we solve your personal problems. I'll drop a link in the description for both the forms for classes as well as Patreon, and I'll link the paper for May-June 2024, paper 1-2 up here when I solve it, and the paper for May-June 2024, paper 1-3 down here. And remember to hit the subscribe button, and I'll also link the playlist for biology paper 1 MCQ up here. Okay guys, see you, take care, bye bye.